How's everyone this morning? Good.
Good.
Nice. All right. Don't know what happened to the rest of the group, but it is 9:30. So let's start with Neuroccino on this Monday morning. Today we have Lilith present a recent paper from the Nature group on the long segment of the arcuate in chimps. Lilith, take it away.
Yeah, so I'm sharing the link in the chat and the screen. You should see now it well. Okay. Um, let's start first. I'm super excited to share this paper with you. This paper was published uh, in April of this year by Yannick Becker in Nature Communications. I chose this paper mainly for three reasons. First, I think this is very rare study because it utilizes the high resolution imaging in chimpanzee brain. The second one is that their dataset comprise of both wild and captive chimpanzee, which is also, I think, adds to this rarity of this paper. And the third one is because it makes quite a bold claim that they found evidence for potential structural precursor of a language network. So I was really curious to know what is their position in this. And and funny thing is that they also show the reviewers' comments. And I think before, on their first attempt, there was just as structural precursor, and then they make it less stronger, the argument, and make it potential. So yeah, let's go next and start to see what they have. So first, I mean, you all are familiar, I think, is that in human brain we have this structure, white matter tract, called arcuate fasciculus, both in the left and in the right hemisphere. Here they are talking about arcuate fasciculus as long segment, so the only connection from frontal to temporal, posterior temporal lobe. Okay, the but within that long segment, they are discussing it in the context of two branches. So the one branch that goes from frontal to superior temporal gyrus, and the one that goes to the middle. So if we go to this picture, maybe it will be easier to remember. So we have the one that goes to STG and one that goes to MTG. And this is all is like basically long segment of the arcuate. Um, so what is the background story for the chimpanzee brain? So it seems that before we thought that the chimpanzee brain doesn't have the connection to the middle temporal gyrus, so they have only the one that connects to the superior temporal gyrus. This is the first thing. And second thing is that this connection to the superior temporal gyrus is actually symmetrical. So there is no lateralization. And we thought that, well, this might this MTG connection tells us why we can speak, have language, while chimpanzee cannot have, they don't have language. And also this lateralization happened because of the specialization of the language to the left hemisphere, which is, yeah, obviously can be also debatable in some points, but this is the main idea, okay? And they wanted to re-examine this if actually chimpanzee brain doesn't have the MTG connection and if there is lateralization or not. Okay, so they had 20 individuals of chimpanzee like brains. It's all post-mortem. And I think the high resolution was like 9.4 Tesla. I will double check later, but yeah. And it's pretty balanced in sex. And also to compare it with humans, they took 20 sex-matched healthy human participants as well. They did 3 types of tractography. One was the manual, deterministic. 2 others were probabilistic. Tractography, in which sense to others, is that they used here both in probabilistic. So this one was manual, and this one was with predefined atlas masks. So first thing what they found is that if we do a deterministic one, okay, in that case we see that that there are only like 8 individual chimpanzee brain that were present, that this connection to MTG was present. But when we do probabilistic one, both in these like anatomical cases, the deterministic tractography with predefined atlas, in both cases we see that we have those connection in all individuals. Where is it? Yeah. IF-MTG connection was detected in all 20 individuals using probabilistic tractography. And if you do deterministic one with also predefined atlas, you see the same results as the probabilistic tractography. This next, they looked at the strength of the connectivity. And what they found that this connection to STG was stronger than MTG in chimpanzee brain, while this pattern was reversed for the humans. So in both cases, we have this connection, but STG was stronger in chimpanzee and then MTG, while in humans was the reversed. The next interesting result is that they looked at the hemispheric specialization, like lateralization. And in this place, they looked at those brains that had the values with a specific threshold, basically. So the ones lower than 0.025 was considered to be left lateralized. If more than 0.025, then it's right lateralized. So anyway, using this type of technique method, they found out that, remember that we thought that in chimpanzee brain, we thought that there is no lateralization of STG. So, well, it seems that in some individuals, you can find those lateralization. So they are either were left lateralized or right lateralized. Although 10 plus 8, 18, so I don't know where's the last one, probably it's just was bilateral. But and then we have the human brains, and in human brains, the majority were left lateralized, both for STG and MTG. Basically, when they did also the group analysis, they show that left lateralization in chimpanzees tract is not statistically different from the human left lateralization. So chimpanzee brain and human brain, they are both have similar lateralization to the left. Boom. And then they're like, okay, but let's look at the differences between wild and zoo comparison, like the ones that were in captivity and the one that were in wild, the chimpanzees. Unfortunately, they don't have a big sample size to drive strong conclusions on this, but some preliminary descriptive information, you can see that only one zoo house chimpanzee, okay, was not left lateralized. So 8 out of 9 were all left lateralized. While in wild, chimpanzee brains, there were more variability. So there were some right lateralized and some left, while only one of this small sample appeared to be not left lateralized. So later they are in discussion talking about, well, maybe this, because those chimpanzees that are in, in captivity, in zoo, they have more human interactions, and there is this training called do as I do, imitation training, and they thought, although they don't have yet like strong statistical evidence for this, but they just hypothesized, so maybe this interaction drives this plastic change and make it their brain more similar to human in the sense that it becomes more left lateralized. But this is really at the moment at a more like theoretical level, this conclusion, interpretation. Well, let's look at a little bit to the pictures. They're beautiful. Here they are showing that in the blue we have this Arcuate fasciculus. And also you can see on this contrast really well the difference. But I have a question here because, well, I'm not really familiar with the chimpanzee brain, but I would guess that this is Sylvian Fisher, then this is superior temporal gyrus, and then this should be middle one. And at least in this section, honestly, I don't see that it goes to that, but maybe it's just either I don't understand their anatomy or it's just a wrong picture to demonstrate this connection to middle temporal gyrus. Yeah, and here they are showing that, well, it goes to MTG. And this is, yeah. Another thing that they did, and they are not showing it in this main paper, is in supplementary materials that they also used a publicly available dataset for macaque brain. And they did also the analysis. And they showed that, well, they don't have the connection to MTG, but I mean, basically that's because I don't think they have MTG. So it's just, or it is merged with inferior temporal gyrus. But yeah, so they also did this comparison to the macaque brain. And then if you look here, this is so basically if we look like in summary, we thought that chimpanzee doesn't have the MTG connections. So they do. Second, we thought that there is no lateralization of STG. There is. And also of MTG there is. The only difference from the human brain in this study they found is this reverse ratio of the strength of the connectivity. So, for example, again, in this case, STG is stronger in chimpanzee brain, while this pattern is basically reverse for the MTG. But for me, it was also interesting to see that there is larger spread, so there is more variability in MTG. So there are like actually some, chimpanzee brain connectivity that are pretty stronger and at the level of this. So I was wondering to do like more individual level also comparison of see if there are some chimpanzee that's showing exactly same reverse ratio as humans, because it seems that there are some values higher than those. Let's see what I can say. Oh, well, maybe I can show also these supplementary materials. They are, they are talking about their questions or. yeah. Well, I was looking at the chat, sorry. But yeah, if we look honestly to this, I don't know if this is really well made dissections. And then if you look at this situation, the threshold at 5, I mean, I don't know. But then if you do this, that looks something very similar to what we have. Usually what we see in this case, you see that it doesn't go to the middle temporal gyrus. So yeah, I was wondering to see what is your opinion in this. Yeah. Another thing, even if the dissections are right, and even if we have this connectivity to MTG, I still cannot be convinced that this can be a precursor of language network. I mean, this would make more localizationistic proposal that, okay, MTG is, what is it here? Yeah, like STG connection is the repetition, and the MTG connection is lexical, semantic, and syntactic mapping. So like, and if there is no MTG, so there is no these processes, and if there is MTG, so there is, so which kind of makes this tract that branch of long segment very functional specific, and if it appears in the brain, then it means that that might be the precursor. So I don't know. This part was a little the functional relevance. I mean, I think it's very valuable study, but I think we need more studies like this to understand what is actually the precursor of language. Another thing, the last note that I will tell you, and then we can go through 2 questions, is, well, I didn't know this, but it seems that chimpanzees' goal-directed vocalization and gesture suggest possible precursors of syntax and semantic, with some having specific intentional meaning. So, in contrast to monkey vocal repertoires, the vocal utterance of chimpanzee contains structural properties where units are either emitted singly or are routinely and flexibly combined into longer utterances, some of which are compositional. So I didn't know this information. I thought might be also interesting that I share it with you. But yeah, this is a very short study, but with a lot of information. But I was wondering, what is your opinion on this? So thank you.
Thank you, Lilith. Any questions? I see a physical hand up, I think, Anna.
Sorry, hi. Yeah, because never mind, I couldn't react because my window was a little bit, never mind. Thanks so much, Lilith. Thanks so much. I saw the paper, I wanted to read it, and now it came out in the Neuroccino. I'm really glad. I was wondering, so one of the main findings was basically the contra, like, that in humans, the MTG, no, is stronger, right? And
yeah, here.
so like exactly that figure. Do they deliver any explanations about that? Like, do they discuss discuss what it is?
Yeah. So the main thing is that before we thought that the absence of MTG, the lack of MTG in chimpanzees,
is responsible for them not being able
to speak language. And here they're saying, well, that's not true. There is, but the difference is this change of the strength. And they say that human-specific arcuate morphology likely evolved by strengthening an existent connection in the brain rather than arising the novo. So I mean, they're just saying, okay, this is evolution, evolutionary, there is morphological changes of these structures. And instead of saying, well, there is no MTG, so there is no language, and they say, okay, there is MTG and it is less strong than human one, so that is why it is a precursor of language. But again, this connection to the function was, to me, not very convincing. But still, I mean,
Can I jump in on that as well? So along the same lines, in the Croxon paper in 2018, we showed that it makes a difference if you look at the absolute difference between non-human primates and humans and the relative difference. I was wondering if they corrected the size of the STG, MTG part of the by brain volume.
Yeah, I think they did it. Let me check. This is in the methods.
But also they only use 5 streamlines, right? Or was it, did I understand correctly?
No, they are just showing how it changes in supplementary materials, how it changes.
Okay.
So because if you do, for example, 100 streamlines, first of all, it just disappears in the MTG. This is the bottom is the MTG connection. It just disappears. And 30 streamlines, well, I don't know. But
So
then if you use 10
results-
streamlines, then you have like some, like just the whole brain is basically becomes Arcuate. So,
So what are their main results based on? On how many streamlines do they specify that?
let's check. Okay.
Interesting to see it in direct comparison, like the human brains and then the chimp brains, and to see basically how massive the connections in the human brain are for like the arcuate, and barely visible in the chimp brain.
Well, if you spot information on this, let me know. I'm trying to find it too.
Do the control F.
Yeah, but what? Yeah, streamlines. That's the control of streamlines.
Not smarty pants. I didn't want to, you know, be a smarty pants. I'm just used to giving Michelle always technical advice on those shortcuts and the modern stuff we're using.
That's fine. We can check those questions later.
Yeah, it's a little bit detailed, but yeah.
Sorry, Anna, were you done with your questions?
Sorry, I'm done. I'm gonna.
Sorry, like I think based on this image, it is more similar to this one. So I guess they use 10 streamlines for this. And for the human one,
I guess that's the threshold we see
30.
on the figure. Yeah.
Yeah. So for the chimp, they use this one. And for the humans, they used, I guess, this one, 100. Let me see. Yeah, seems similar. For the image. So, but the calculation exactly, I need to double check the supplementary materials, how they did it.
And is volume the only measurement they extracted? As in voxels intersected, or do we have more measurements? Any indices, anything?
No, in the main text, also like the strength, I didn't know. I mean, it's not written what do they mean by strength in this case. They would say that it's stronger. So they are not mentioning in the main text, in the methods. Yeah.
Okay, so mainly volume-based, at least for the main finding. Then a couple more questions that I would have on it is, so number one, if I understood you correct, they compared deterministic and probabilistic, and they compared manual in deterministic and atlas-based in deterministic and probabilistic. And you, just clarifying that I got this right, but you said earlier that if they used manual dissection deterministic tractography, they weren't able to reconstruct the arcuate in all hemispheres, right?
They were, no, if you do deterministic manual, you
Yeah.
find this MTG connection only in 8 individuals.
But if you use an atlas, you find it in most...
all, whether it is probabilistic or deterministic.
Right. Do they share their manual regions in that case? Because if the atlas can find it, but the manual dissections, wondering where the manual...
Do you see my screen?
Yeah, currently at the results.
Let me show you then something else. Let's go over here. Wait, sorry, fast, share. You see something else, right?
Yeah, FSL.
Yes. So they actually share all their data, the human, chimpanzee, and tracking recipes. I guess this is what you meant. Not this, but like, I don't know. So for example, exclusion, frontal left.
Those are the manual deterministic regions, and that's what I meant. But I guess they shared them somewhere.
Well, this is the only folder that they shared. And I think, I don't know.
Because
yeah.
seed points towards probabilistic, not deterministic.
Yeah, so then, and this is the chimp one. One of the individuals that I found, just to visualize.
Cool. Okay, so that's clarified. Thank you. The other question that I have is that you mentioned that they hypothesized this more lateralized arcuate in the zoo house chimps may be based on exposure to human language. So in a way that experience, environment is driving that lateralization towards the left. Now, I was just wondering how to place that in the literature, because very preliminary genetic data or genetic analysis seem to sorry for stealing your question, Abby. maybe my ending is different than the question you had. But very preliminary genetic analysis in tractography seems to point towards the long segment being the one segment that's most driven by genetics, and the indirect segments, so the anterior and posterior, being more driven by environmental and exposure. So I was wondering if there's any comment on that, and I happily hand over to Evie if you wanted to add to that.
No, there is no comment, but then I might think the authors would argue that maybe at the level of chimp brain, there is still a plasticity of the long arcuate, maybe in the same way as in our human brain, there is more plasticity in some segments of the arcuate rather than being strongly genetically driven. So maybe they would say at the level of chimp, there is still some flexibility in the long arcuate, but yeah, I don't know. They don't comment on this. But for the curiosity, I also checked the dolphin brain, and there is a tractography of the dolphins. And in that one, they said that all the tracks were left lateralized except arcuate and some segment of SLF to the right. So so I don't know, it's just information. I cannot connect it with any function. And the same way here, okay, they are left lateralized. Okay, there is some little branch going to MTG. What does it say actually about the language as being as a substrate for a language precursor? So yeah, I don't know.
Yes, so and following on this question, so yeah, thank you, Steph, for stealing my. But yours was deeper in the variability between genetic and environment, and so mine was more about So they look at wild chimpanzees and zoo housed chimpanzees, and I was wondering, looking at only at the genetics. So when you look at zoos, chimpanzees, maybe there are less variability in genetics because they maybe reproduce each other. So when you look at your plots, they looks all the zoo house chimpanzees looks left lateralized, and there is more variability. So I was wondering if they discuss that, because if you select chimpanzee in the wild, yeah, you will get more more variance, more diversified population of chimpanzees. So in human, we check for a lot of stuff on that. So I was wondering if they did that.
No. I don't think they did it. I don't remember this. So, but I think this is really great point. I mean, and it would have been really nice discussion paragraph for them as well.
Yeah.
Yeah.
No,
That's
it's true. They don't have a large number of
no,
chimpanzees, so it's complicated.
yeah, but also some background information on those captive
Yeah.
ones would be even a little bit more useful to
And yeah,
manage to interpret.
and even in the plots, so when you were showing the box plots, yeah, it would have been interesting to. So you saw when at the end of the paper when they show humans and chimpanzees, it would have been great to distinguish these two populations. But
Yeah. So they said that because when they did comparison with humans, the STG, MTG, these things like the strength,
Mm.
as I understand, they used it like both groups in one, and this left lateralization was mainly driven off from
Yeah,
those, those zoo chimps.
yeah,
So that's why they try to then do two group comparisons, but then it's just to 8, 9 brains. So that's why it's like more on hypothesis level.
okay. Thank you.
Thank you.
Another question from my end is the novelty of these findings. Do they comment on the previous studies, like the Rilling paper from, what was it, 2008? They already showed some connection to the MTG. And there have been other papers, at least two that I'm aware of in the meantime.
So yeah,
Comment on the novelty?
I don't know if they are commenting, they are referring to that paper, but there is mentioning that there was already a study that used 4 chimbrains showing that there is might be MTG connection, but then other larger studies they showed that there is no. So that was kind of like it's still debated. So and they said, okay, we're gonna take 20 and do the high resolution imaging. So that's the novelty that they are kind of clearing up things in the sense that what is in the end is there or there is no, and also like there was 4 chimbrains and maybe they used their manual deterministic and there was nothing dissected.
Okay. That brings me to my final question. How did
Yeah.
they actually validate it? Because what you've showed, if I'm not mistaken, is tractography and FA maps. But they go back to exactly the same raw data, which is diffusion weighted imaging data. So the FA and the tractography are not validations of each other. They go back to the same data. Is there any validation step in this?
They did not mention that they did post-mortem dissections. Like
it doesn't have to be post-mortem. It can be PLI tracing something to validate the diffusion.
I had no information about this in this paper. So in supplementary materials, I don't think they have it either. Yeah.
Right. So we still don't
Right.
have a definitive answer on the question of whether the long segment exists.
No, the long segment kind of exists, but
where it goes, the extent of it, and
yeah.
And its function. The thing that is interesting to me is if it is there to that extent, our language network is not unique. So how do we interpret that going forward?
Well, I don't know.
Neither do I. No answer just yet. But that's all the time we have today. Thanks for joining, Neuroccino. And
Yeah, thank you.
science. See you next week. Bye.
Thank you. Bye-bye.
Bye.
Bye.
Bye-bye.